Okay, this is part 2 for Lab 10. I want to go through the lab report questions and provide some more context so that you are better equipped to answer the questions. And I know that my first set of slides lacked some essential information, and so I want to go through that with you now. To do that, I want to start by reviewing the central dogma of molecular biology, which then obviously has critical applications to microbiology and how organisms work. So the central dogma refers to the flow of genetic information from DNA to RNA to proteins. And so I want to look at this particular sequence. This is from the first question of the lab report. It's the same sequence. I've just taken out all this middle part, so I just took these first few sequences and these last few sequences, or I'm sorry, nucleotides, and then I just deleted everything in the middle, so it's a little bit easier to look at. So I provided you the coding strand of the sequence that we're looking at. So the coding strand is directly converted to it is the template. Okay, sorry, I'm using bad words here. The coding strand provides the nucleotide codons that provide the amino acid sequence. So remember, in groups of 3, they are converted to amino acids. And so what that looks like is ATG becomes an amino acid, TTT becomes an amino acid, GTT becomes an amino acid. And so if I want to know the amino acid sequence, I look at the coding strand, and that tells me the sequence of amino acids that I will have. So ATG becomes methionine, TTT becomes phenylalanine, and GTT becomes valine. Because DNA is double-stranded, I will also have what's called the template strand. So the coding strand is the code of amino acid sequences. The template strand is what is used by the polymerase to make mRNA. That is then converted into amino acids. So if I know the sequence of the coding strand, I don't have to know the sequence of the template strand. Excuse me. I do not have the coronavirus. I am choking on granola. Okay, so if I know the coding strand, because the template strand is going to be the reverse complement, if I have one of the DNA sequences, I do not need the other one. I can infer it. So if I have an A, I know that I'm going to have a T on the opposite side. If I have a T, I know that I'm going to have an A. If I have a G, I know that I'm going to have a C, etc., etc., etc. And so with that in mind, here is the template strand. Again, it's predictable because it's the central dogma. It always follows this how do I say? Like it will always it is predictable, and it always follows this base pairing. Okay, so again, if I look at the coding strand, I know that ATG will give me one amino acid, TTT will give me another amino acid, and then GTT will give me another amino acid. And so because it's the coding strand, I know that the product is going to be a particular amino acid sequence. If I want to get this, so methionine, phenylalanine, valine is what that is. If I want, by following the central dogma, I know that I have to get from DNA to RNA to amino acid. So if I want to produce an RNA molecule that gives me this same amino acid sequence, how can I do that? So let's look at the wrong way to do it first, and that is by converting the coding sequence to RNA. If I convert the coding sequence to RNA, complement RNA, so I get U, A, C, and then I'll get 3 A's, and then I'll get C, A. Okay, so this RNA sequence, what will be the amino acid sequence if I translate that? It will be this amino acid sequence, which is very clearly not the same thing as MFV. So I know that if I use the coding strand to make RNA, it will give me the incorrect sequence. Therefore, I must use the template strand, and that is why it is so named, is because it's the template for creating the mRNA. So let's convert this now to mRNA. So I got TAC, so that will be AUG, and then I get 3 U's, and then I get CAA, which will be GUU. Okay, if you look at this RNA sequence is the RNA copy of the coding strand. Because now if I translate this, it will give me the predicted amino acid sequence, which is MFV. So that's what this looks like. So AUG, UUU, GUU. And again, to get the correct mRNA, and I didn't put an M here, to get the correct mRNA sequence, I have to transcribe the template strand to mRNA. And the mRNA will match the coding strand. Okay, so with that in mind, let's talk about RNA viruses. If it's positive sense RNA, it can be directly converted to protein. In other words, the positive sense RNA is the exact same molecule as mRNA. So it skips a step. Rather than having a coding strand having to convert to mRNA, it is already there. And remember, I made the mRNA from the template strand. So mRNA will be essentially the same sequence as the coding strand, except for the fact that the T's in the coding strand will be used in the mRNA or the positive sense RNA strand. The negative sense RNA has to be converted from RNA to RNA, which can then be converted to protein. So if I want, as I mentioned in question 1, so I said that the sequence has been reverse transcribed from RNA to DNA. So because, again, it is a positive sense RNA strand, I know that it's the same thing as mRNA. I know I'm saying this so many times over, but it's absolutely essential to understand. And as we continue working with DNA and protein sequences in the next couple of weeks, it will be absolutely critical that this information is solidified. So if I have a positive sense RNA strand, it will look like the coding strand. And so you'll see here everything's the same except the T's have been converted to U's because, again, RNA uses uracil rather than thymine. Okay. So that is the central dogma. Now, going back to question 1, so I'm asking you to make primers for that sequence, okay? And so in my previous slide, I included the Shindell-Garnu sequence and some non-coding region before and after the gene. This is not the case for this question. It starts exactly on the start codon, and it ends exactly on the end codon, or the stop codon. So the primers will only cover the gene. There's no Shindell-Garnu sequence, so it's going to start exactly right here and do, as I said, 12 to 15 nucleotides long. So let's do a practice problem of that. This is a similar sequence. It is not the same sequence because I didn't want to give you the answer. So here is a DNA sequence. So again, if this were an RNA, a positive sense RNA sequence, I have converted it to DNA. And so the positive sense mRNA or the positive sense RNA will be the same sequence as the coding strand, just with thymines instead of uracils. And then again, the template strand is inferred from that sequence. And so that is what you are looking at right here. Okay, so let's denature the DNA to make our primers. So the first thing that we have to consider is, again, the directionality of the DNA. So if I want to synthesize, I can only synthesize in 5' to 3', which means I have to read in 3' to 5'. So if I'm going to synthesize a complement of the coding strand, I have to start here and move in this direction. This direction. If I want to synthesize a complement of the template strand, I start here and move in this direction. Which means, where do the primers have to bind? For the coding strand, it has to bind here so that I can again move in this direction. And if I want to, if I have a primer binding the template strand, I have to bind here at this sequence so that I can synthesize in this direction. Read from 3' to 5', synthesize from 5' to 3'. So how do I get a segment of DNA or how do I get a primer to match this segment of DNA? Well, for a primer to work, it has to be the reverse complement. What is the reverse complement of this sequence? It's this sequence right here. So if I want this primer, all I have to do is take the coding strand segment, 12 to 15 nucleotides, and that will give me the primer that binds the template strand. So this is called the forward primer because, again, this is the beginning of the gene according to the coding strand. It begins at this ATG and continues in this direction. And so it's the forward primer because it will synthesize the reverse complement in this direction. Similarly, if I want to create a reverse primer, I need to bind in this segment. How do I bind this segment? Again, look for the reverse complement. Where can I find the reverse complement? Well, it's going to be this sequence. And so now if I have a primer binding here, it will synthesize 5' to 3' and move in this direction. So now I can go here and I can also go here, and I'll end up with 2 double-stranded DNA segments. So now if I take this away, and I'm only giving you the coding sequence now, how do I design the primers? Well, the forward primer has to bind the complement of the coding sequence, also known as the template strand. And the reverse primer has to bind the coding strand at the end of the gene, and then this is at the beginning. So again, if I want to bind the beginning, it has to be the complement of the template strand, which happens to be the exact sequence that I've given you. So this will be the forward primer. Again, it's not the correct length, but you get the idea. And which end will be the 5' end? Consider where it's binding on the template strand and the direction that it has to face. So it's synthesizing 5' to 3', so it will be going this direction, which means this is the 5'. For the reverse primer, I need it to bind the coding strand, so binding the coding strand at the end of the gene. So I take the end of the gene, and how do I bind this exact sequence? Well, I need the reverse complement, which is going to be, here, we'll just write it. We'll write out these last few again. So we get TAG, TT, G, AATT. And so this will be the reverse primer, but which way is it facing? It's actually backwards, so this is the 5' side. So now if you're looking at this sequence from question number 1, I've given you the coding strand. It's in DNA format. So to find the forward primer, what do you have to do? This. To find the reverse primer, what do you have to do? This. Okay, so I hope that makes a little bit more sense. For number 2 on this, how do you find the length of your expected product? I gave you the base pair numbers, and so I will let you figure that out. For number 3, this is also, I didn't explicitly explain how to, the answer to this question in my slides. But if you get a PCR product that is not the length expected, assume that you did everything absolutely correctly, but it still came out a different length than what you expected. Assume that there was a genetic cause to that. So something happened in the gene that made it shorter than expected. So what could happen that would cause that? Let's go over PCR. Sorry, not PCR. Gel electrophoresis. Just so you can see again what this looks like. So assume again, so I have a tube. This is my PCR product, and I ran the reaction already, and I have now millions of copies of my gene segment, and I want to check it on a gel to make sure that it's the expected length. So what I will do is I would take this pipette and I would pull from the PCR, and then I would put them into these wells. So over on this side is top looking down, and then this side is looking from the side. So these wells are actual indentations in the gel they're empty space that you can fill. So if I fill it, this is what it looks like. Okay, remember, DNA is negatively charged, and so if I run an electric current through the gel, and it is under liquid, so it is submerged completely in a salt solution, so that electricity transfers through it. So if I run electricity, the DNA will move towards the positive electrode, so the cathode, because again, DNA negatively charged will be attracted to the positive charge. And so I've labeled both sets of gels with the anode and the cathode so you can see that. All right. So now let's run the gel. So now I filled all of the wells. You can see up here I filled these 5 wells with sample, and then these 5 wells with the ladder. Okay, turn on the electricity, and it literally gets pushed through the gel. And the gel is a complex or a matrix that the DNA has to maneuver through. I sort of messed up. This should be present on all 5 lanes right here. I didn't include that, but you get the idea. So the DNA segments are pushed through the gel downward. Okay. And so that is what's happening on gel electrophoresis. The purpose of this is to check the length of your PCR product. So again, if I say that this is 750 base pairs, is that the expected length? That's going to depend on the target sequence, the target gene of your PCR product. In question number 3, it is not, but in other circumstances, it generally, PCR usually works out pretty well. Okay, so PCR and gel electrophoresis are how you check the length of a gel to see if your product is present. You have to actually visualize it, again, using UV light to see this present on the gel. qPCR, I want to completely redo this because it probably didn't make any sense at all without my annotations. So qPCR is basically PCR 2.0. It saves a step. Rather than having to take the PCR product, put it in a pipette, put it in the wells, and run it through the gel on gel electrophoresis, QRT-PCR, sorry, I said that wrong, QRT-PCR gives you instant results without having to run it through a gel. It basically, as the PCR is occurring, there is a spectrophotometer looking for fluorescence. And so these lines that you see are fluorescence levels. The threshold is the minimum amount of fluorescence required for the spectrophotometer to pick up the fluorescence to see if it's glowing or not. So what it will do is, remember, because it's exponential, it doubles every round. So N is the number of cycles. So number of cycles. So whatever my starting product is, assuming I start with one molecule of DNA at N at the first cycle, so N of 1, it doubles. So 2 to 1 is 2. If I run 2 cycles, then it's times 4. This is just a little equation of how to get that. So 1 is my starting molecule, and then I multiply it by doubling it every time. Okay, so looking at qPCR, I can see that, like, let's look at this green line for a moment. So this is the number of cycles, so 5, 10, 15, 20, 25. And then this, the fluorescence value, don't worry about that. Just know that at a certain level, let's say, as I said in my previous one, so at 10 to the 6th, so at 1,000,000 copies, there was enough fluorescent molecule, there was enough DNA to bind the fluorescence molecule to glow, and the spectrophotometer will pick that up. So at how many cycles is this? So this is about, let's say this is 12 cycles. The green sample has enough DNA, so it has 1,000,000 copies of DNA to reach the threshold value. So if I know there's 1,000,000 copies at 12 cycles, I know that 11 cycles, that there were 500,000. At 10 cycles, there were going to be 250,000. At 9 cycles, there are going to be 125,000. Backwards, you can calculate the starting amount of DNA that you had of the green sample. Now let's look at the teal sample. So this one, let's say it took exactly 25 cycles to reach 1,000,000 copies of DNA. So remember, 10 to the 6th copies of DNA, and it took 25 cycles. So if I know that it doubled every time, at 25 cycles, I have 1,000,000. At 24 cycles, I have 500,000. At 23 cycles, I have 250,000. And so you can also calculate, again, going in reverse, meaning that at the first cycle, the teal sample had fewer molecules of DNA because it took twice as many cycles to reach the threshold value of 1,000,000 to fluoresce. So just to reiterate, QRT-PCR, so quantitative real-time PCR, is PCR 2.0 because rather than having to run my sample now on a gel, I can just quantify it as it's occurring and see how many samples I had to start with. So this is useful because I can figure out the amount. Well, I can test for the presence of DNA because if, like, let's say again, if I was targeting like a beta lactamase gene, and I got a bunch of bacterial samples, let's say I had 20 samples. Well, if I have so I have green, red, black, pink, and teal. So 5 of the 20 samples fluoresced, which means 5 of those samples had the beta lactamase gene in them. The other 15 samples did not. So rather than having to run on a gel and nothing appearing in the lane, I can say, okay, 15 of these samples didn't give me any fluorescence at all. At any amount of cycles, even up to 45 cycles, nothing fluoresced, which means that there was no starting copy. Because remember, if you're doubling, even just starting with 45 times, you're going to have a very, very large number, and it will show up on this assay. Versus, like, in the green sample, it showed up very early on after only 12 cycles, which means I had many copies, like, it just means that the gene, the beta lactamase gene, was present. And same thing with the red, the black, the pink, and the teal samples. So if I'm looking at, let's look at number 6. So how can I use that to test somebody to see if they have a coronavirus present in their system? I take a sample, I run qPCR on it, and let's say I use the spike protein gene. So I target this specific gene sequence, and if it fluoresces at all, then I can say, hey, they have coronavirus because I saw fluorescence, and that means that the gene was present. Okay, so that is QRT-PCR, and that should also help you answer number 7. Let's jump back to number 4. So this, I want you to think back to the GFP lab with the the green fluorescent protein plasmid. What I'm asking for here is what is required on this plasmid. It's a question similar to like what our quiz was. What is required on the plasmid to make it functional, to actually express the protein? So how do I tell the cell to keep it? How do I induce protein expression? How do I make sure that all cells have a copy of the plasmid? That is what that is asking for. Okay, and then the last one we'll talk about is number 5. So this is asking about lytic versus retroviruses. In what we talked about phages, so we talked about lytic phages and lysogenic phages. Consider both of these life cycles independently, what happens to the bacteria on each. With the lytic cycle, if I get infected with a bacteria, if I'm a bacteria and I get infected with a lytic phage, what immediately happens? The phage proteins get expressed, they enumerate inside of the cell and burst the host cell, and it dies. In a lysogenic phage, what happens? It inserts the phage DNA into the host genome, and it proliferates as part of the genome, doesn't express phage proteins until it gets stressed out. And so what would that look like now if I have that happening in a human cell? And if I have a retrovirus, so lysogenic is for bacteria, retrovirus is for eukaryotic cells. So if I have that happening in one of my cells, what would happen? So what's an example of a retrovirus of a human cell? Think about that for a second. A good example of a retrovirus is HIV. It inserts its genome into T cells, which are immune cells, or the chickenpox virus, pox virus, is a retrovirus, and so that is why you can get shingles later on in life if you've had the chickenpox as a child. And that is because the genome of the pox virus is inserted into neuronal cells and expressed under stressful conditions. So it's exactly like lysogenic phages, just in our own cells. So with that in mind, is SARS a lytic virus or a retrovirus? How do you know the answer to that? And which one do you think is more lethal? And with this last one, there is not necessarily a correct answer, but I want to know what you think and why. Okay, I hope that this has answered all of your questions regarding the lab report. I will still be available. I will also be available Monday. I mean, I'm always available, but I will be extra attentive to emails on Monday because this is due on Tuesday, and so because that's the day before it's due, I'll make sure to be available that day as well. So again, please let me know if you have questions or if you would like further clarification on anything, and stay healthy.